So welcome everyone. Thank you for joining the UQDI Fellowship Program, which 10x Genomics, together with Millennium Sciences, has put together. It's an incredible opportunity to develop single cell workflows locally and at UQDI and at TRI. So we might kick things off from my end, and I'd like to just start off with an acknowledgement of country. So the University of Queensland acknowledges the traditional owners and their custodians of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connection to country. We recognize their valuable contributions to Australian and global society. Today we've got a stellar lineup of presenters, and I'd like to first introduce Jeff McDermott. And Jeff's the scientific and technology advisor at 10x Genomics. We've also got Daniel Lugolensky from 10x Genomics. He's a commercial lead. We probably see Dan through the TRI often, so he's a familiar face to all of us. And we've got some other DI researchers. There's also Gabrielle Bells, who will be talking to us about some of her work too. So I'll probably hand over to Jeff. Please take it away.
Thank you so much for having me here today. I'm a science and technology advisor with 10x Genomics. Before we begin, I just wanted to set the stage a little by going over what to expect over the next 15 minutes or so. We'll begin with a very quick mention of our experience event, which will be happening right after this meeting, and then some quick background on what the UQDI Fellowship entails. Then we'll head into a general product portfolio introduction, and after this, take a deeper dive into our Chromium single cell and Visium spatial transcriptomic solutions. We'll step through what each of these assay types has to offer, but given the short amount of time today relative to just how many products we have, won't be providing much data. Instead, the main goal of today is really to introduce you all to the assay capabilities of what we offer at 10x Genomics, which of course you now have access to through the UQDI Fellowship. So we begin today by quickly mentioning our upcoming experience event. As I said, it's actually straight after this meeting. For those of you who are unfamiliar with this virtual event, I will be highlighting our latest developments and give you a sneak peek of what's going to be available in 2022. Unfortunately, I don't have time today to cover all the new products that we're announcing, so I highly encourage you to attend. But if you can't, then please just reach out. I can provide the recording of the slides. The next piece of housekeeping I wanted to cover before reviewing our product portfolio was to cover what the fellowship program actually entails. So the fellowship program is open to graduate students and early to mid-career researchers with no pre-existing experience using 10x Genomics technology. To apply, you simply submit a short project abstract of no more than 250 words, which then gets reviewed. And upon acceptance into the program, you'll get all the benefits that are listed here, in addition to a few things that are not listed. The key things to point out from this list are discounts to the 10x Genomics reagents, which of course everybody loves. But in my mind, the most important feature of the program is the support that we offer right from when you're conceptualizing your project through to the actual data analysis. It's really our job to mentor you through the entire process and ensure that your first experiences with our products result in successful outcomes and really great data. But now let's focus back onto reviewing just what products we actually offer at 10x Genomics to give you an idea about how they can be used to drive your research forward. And we'll begin with a general introduction into our product portfolio. And our assays can be broadly associated with one of three major technology platforms. So let's briefly step through each of these, beginning with the Chromium single cell platform. And as you can see, this consists of an instrument called the Chromium controller, which is about the size of a shoebox and fits on your lab bench. Into this instrument, you feed these disposable microfluidic chips, and then onto those chips, you can run one of four main assay types. And we'll dive deeper into these on the next few slides. But irrespective of the target analytes, the key output from these assays is the ability to characterize cellular heterogeneity. I do also want to highlight the sample format that's required for this platform, which is a suspension of single cells or single nuclei. So this means that unless you're already working with cells in suspension, say such as PBMCs, your tissues or organoids will need to undergo some form of dissociation to generate a clean, viable suspension of single cells. The second major technology platform is Visium spatial transcriptomics. Unlike the Chromium single cell platform, there's actually no instrument involved here. Now you're just dealing with this consumable, which is a glass slide that contains barcoded arrays on its surface. Also, unlike the Chromium platform, this technology now uses whole tissue slices as the input rather than a suspension of single cells. Here, the section tissue is placed on the slide and transcripts are diffused from within the tissue section down to the surface of the slide, where they can be subsequently barcoded. And following sequencing, we identify what the transcript is and then use this barcode sequence that's appended to the transcript to place it back into a 2D grid, giving spatial context to the transcript and ultimately a spatially resolved molecular profile of your tissue. We'll cover Visium in more detail in the second half of this presentation. Let's move on to our third major technology platform, which is our in situ product, or as we've now named it, Xenium. I just have to say that this piece of technology is something that I personally find super mind-blowing. Just like the Visium platform, here the input into this workflow will be tissue sections. However, on this platform, you're now detecting mRNA molecules directly in the tissue itself with cellular or subcellular resolution. So rather than extracting the mRNA out of the tissue, barcoding it on a 2D array, sequencing the barcoded transcript, and using that barcode to place the transcript back into the spatial context, here we're just detecting the presence of the mRNA molecule within the tissue itself. This gives rise to data such as this. Here's an outline of a mouse brain sample showing the entire image tissue. The color is a powerful representation of how rich the data is, and this becomes even more apparent when zooming in and focusing on specific cellular markers. Here at high magnification, you can appreciate the single molecule resolution of the Xenium assay, with each symbol representing an individual RNA molecule. However, Xenium is still under development and isn't expected until later this year. And as such, I won't be talking about Xenium beyond this example today. But if you do want to learn more about it, then please reach out to me, or even better, attend that upcoming experience event straight after this. But for today's presentation, let's refocus our attention on the Chromium single cell and Visium spatial transcriptomic platforms and briefly step through each of these assay types. We'll begin with discussing the Chromium single cell platform and its associated assays. So as I said earlier, this technology platform consists of an instrument called the Chromium controller, which is about the size of a shoebox and fits onto your lab bench, and just shown here on the slide. Into this instrument, you feed these disposable microfluidic chips that are also shown on this slide, and then onto those chips, you can run one of 4 main assay types that are listed here. I want to highlight that although all these assays have a different output, since they all use the same instrument and fundamental microfluidic architecture, then they all broadly have a similar workflow as depicted here. Rather than step through the entire workflow in detail, in the interest of time, I just want to highlight that for all these assays, we have ready-to-use robust workflows, including demonstrator protocols, the various sample types, and turnkey software tools. So if you're new to the world of single cell and want to apply it to your research, but feel intimidated about where to begin, then we have solutions for you across the entire spectrum of the workflow. But let's refocus our attention on those 4 main single cell assay types, and importantly, what the output from each is. And we'll begin with our single cell gene expression kit. So the target analyte from this solution is mRNA, where we're capturing transcriptome-wide 3' digital gene expression from single cells. So that's the core of the assay, but we can actually extend the menu of analytes being detected from a single cell using what we call our feature barcoding technology. You can also detect cell surface proteins in addition to transcriptome-wide gene expression from the exact same single cell. This is achieved by having DNA barcoded antibodies which bind the cell surface protein. If you like, this is akin to FACS, where you use a fluorophore barcoded antibody to detect cell surface proteins. However, now that using a DNA barcode rather than a fluorophore, the number of cell surface proteins that can be multiplexed in our assay is essentially unlimited, as you are not limited by that spectral overlap of dyes in the same way that you are when using FACS. There are, of course, some practical limitations to just how many cell surface proteins you can measure, but you do see commercially available panels of over 100 antibodies specific for different cell surface proteins being routinely used in the literature. Using feature barcoding, you can also detect CRISPR perturbations alongside transcriptome-wide gene expression from the exact same single cell. And in fact, you could even detect cell surface proteins on top of all that too if you wanted to. This works by incorporating a small piece of DNA into the guide RNAs that can be captured with the assay biochemistry. So you get a readout of what the guide RNA was, which tells you about the CRISPR perturbation that occurred in that single cell, but you're also getting a transcriptome-wide snapshot of the functional impact of that perturbation. Let's take a look at our next assay on the Chromium platform, which is our single cell immune profiling assay. Just like the previous example, the core of this assay is detection of mRNA, where we're now capturing whole transcriptome 5' digital gene expression from single cells. Because we're capturing the 5' end of transcripts now rather than the 3' end, we can expand the menu of analytes to now include full-length paired VDJ sequences from T and B cells. And the way this works is that you perform a PCR enrichment step from the whole transcriptome mRNA molecules that were captured from single cells, and you pull out the VDJ sequences. This means there's no computational wizardry here to do the pairing of alpha and beta chains if you're looking at T cells, or heavy and light chains if you're looking at B cells. You're detecting exactly what the cell is making, so the pairing information comes from the cell itself. You can also detect cell surface proteins in addition to transcriptome-wide gene expression and cell surface receptors, all from the exact same single cell. Whilst the oligo barcodes that are added to antibodies for this assay actually are different in structure to the 3' assay that I just showed, meaning that the antibodies are not interchangeable between the assays, the barcoding premise is still the same. And what's super cool is now, on top of all this information you can capture from the exact same single cell, you can also layer on antigen recognition. So just like other feature barcoding assays, this works by applying an oligo barcode indirectly to your antigen of interest. So when the antigen binds a T cell or B cell surface receptor, it remains bound, and this oligo that is present can now be picked up by the assay biochemistry and used to infer the molecular identity of the antigen species present. So from just one assay, you get whole transcriptome digital gene expression, full-length paired VDJ sequences, cell surface protein, and antigen recognition, all from the exact same single cell. We'll switch gears a little now and talk about our single-cell ATAC-seq assay for the Chromium platform. So instead of mRNA, we're now detecting DNA in the form of genome-wide open chromatin regions. This is our first foray into the world of epigenetics, and the open chromatin information reveals areas of active gene transcription, regulatory regions, and binding site motifs. Just like the previous products, which measure gene expression, single-cell ATAC-seq allows you to broadly cluster and identify the different cell types in your population. However, for gene expression, we simply count the presence or absence of a transcript, whereas with our single-cell ATAC-seq assay, we can dive into the regulatory regions that control that gene for a more detailed view. And now I want to cover our final assay on the Chromium platform, which we call Multiomics. The moment we launched our ATAC-seq assay, everyone understood the power of that measurement, but also immediately jumped on, or what if I could get both the epigenetic and whole transcriptome information from the exact same single cell. So this is where this product is born from, our single-cell multiome kit, which is really an amalgamation of our ATAC-seq and 3' gene expression assays. This means that you're measuring both genome-wide open chromatin regions and transcriptome-wide gene expression from the exact same single cell. And what that allows you to do is identification of putative regulatory elements that you can then directly link to a gene of interest. And again, that's all from the exact same single cell. So let's move on from our Chromium single-cell product line to our spatial transcriptomic product line, which again we call Visium. As I said earlier, unlike the single-cell family of products, this technology platform does not actually consist of any instrumentation. Instead, you have this glass-like consumable onto which you place your tissue section, and we'll dive into the specifications of this slide in a minute. There are 2 different assays in the Visium family, and as their name suggests, they're designed to work with different input materials, being either fresh frozen or FFPE tissue. Also, rather than step through the entire workflow in detail, in the interest of time, I just want to highlight that just like our Chromium platform, for both types of Visium assays, we have ready-to-use robust workflows, including demonstrated protocols for various sample types and turnkey software tools. So again, if you're new to the world of spatial transcriptomics and want to apply it to your research, but feel somewhat intimidated about where to begin, then we have solutions for you across the entire spectrum of the workflow. So I just want to quickly cover some technical specifications relating to the Visium slide. Keep in mind that this applies to both the fresh frozen and FFPE versions of the assay. So each slide has 4 capture areas to place the tissue on, where you must fill all 4 capture areas at once when using the slide. Each capture area is 6.5 by 6.5 millimeters in size. And within each of these capture areas, you can see that there are an array of approximately 5,000 barcoded spots, where each spot is 55 microns in diameter. This obviously isn't single-cell resolution, but allows you to capture information from a small handful of cells in any given spot. But stay tuned, as I'll have a note about resolution in a few slides' time. So each spot comprises of a lawn of oligos. An important part of that oligo is a barcode. And even more importantly, each spot contains its own unique barcode. So when we append this barcode to each transcript, we can then use it to place that transcript back into the spatial context of this 2D array. So let's walk through the data types that can extract out of the Visium kit. We'll use this mouse brain tissue for an example and begin by showing that you retain the ability to get a H&E image using Visium. And in fact, you could even do immunofluorescence as well. This means that you can still do all your visual annotation of the tissue. And of course, this is typically where your analysis journey would end. But of course, with Visium now, we're not only getting H&E image of your tissue, but we also capture that underlying molecular information across the entire tissue section in the form of whole transcriptome gene expression. But the real power of the kit comes from when we combine these two data types. And that gives rise to an image in the middle here, where the spatial location of whole transcriptome gene expression can be overlaid on the H&E image. So again, you retain the ability to visually interrogate the tissue, but now you can also query the underlying molecular profile in the form of whole transcriptome gene expression. And finally, I want to finish on that note about resolution, which I mentioned a few slides earlier. One of the biggest requests we've had from our customers since we launched Visium is to increase the assay resolution so that we can push the boundaries of spatial genomics to the single cell level. So coming later this year, we'll have a product that does exactly that, and we call it Visium HD. Here, we've completely reconfigured how we make Visium slides, and in doing so, we can push the resolution from its current form of 55 microns right down to sub 10 microns. And you can see just how impressive such a measurement can be. When looking at this image of a mouse embryo and then zooming into the eye region, you can see how these sub 10 micron spots will overlay with the tissue. We'll offer Visium HD as an end-to-end solution, giving you the same seamless experience and support that you're used to from our other products. So if you want to know more, then please obviously tune into Experience or just reach out to us directly. So with that, I'll say thank you very much for attending today. I hope that you enjoy this very whirlwind tour of our product portfolio. And I really look forward to engaging with many of you in the new UQDI fellowship program. And I'll hand it over to Tom.
We're very lucky today to have Tom Weber join us from the Charlene Knights group at WeHi. And Tom's going to be talking to us about how he's utilizing 10x Genomics technologies to further his research on their breakthrough technology, which they called LOX code. So Tom, I'll pass it to you.
Yeah, thanks, Daniel, for the kind introduction and for the opportunity to talk here today. I'm going to talk about a new technology we have developed in which we call LOX code barcoding technology, and I'm going to discuss how we use this technology to understand a little bit more about the heterogeneity in epiblast fate in mouse embryo. So as the name suggests, LOX code is based on the QuiLOX-P system, and this is site-specific recombinase. And to recombine, it requires 2 LOX-P sites. LOX-P sites are 34 base pair palindromic sequence from P1 bacteriophage. And if Cre binds to those LOX-P sites, DNA loop is formed and which leads to recombination. And as in this example, if the LOX-P sites are pointing in the same orientation, this leads to the excision of the DNA between those LOX-P sites. However, as in this example, if the LOX-P sites are in opposite orientation, pre-binding, loop formation, and recombination leads to the inversion of the DNA in between the 2 LOX-P sites. And so that's really the basic principle of the LOX code technology. But instead of having 2 LOX-P sites, we have 14 LOX-P sites. In between each LOX-P site, we have known DNA sequence. And so when Cre gets active, all those LOX-P sites will start to interact and recombine and creating huge DNA diversity. And so if this sounds a bit complicated, you can think of the LOX code technology as a Rubik's cube inside of the nucleus of the cell. And once Cre becomes active, this Rubik's cube starts to get shuffled into random configuration, which is different to each cell. And this can be used as a DNA barcoding, which is inherited to all the other cells. However, in comparison to the Rubik's cube, which stays the same when you shuffle it, the LOX code cassette can lose elements due to excision during recombination. And so we predicted this different size of the LOX code cassette where different elements have been excised. And we can compute the theoretical barcode diversity of each of those different sizes of the LOX code cassette. And so if we just start with a single element, we have just about 14 barcodes that can be made of it. But if we take the full length cassette, we can make up to 30 billion different combinations. So together with from our postdoc in the lab, Denise Miles, we cloned this construct, which was really challenging because of the many repeats in this construct and the palindromic nature of the LOXP sites. And so we managed to clone up to 14 LOXP sites in a row, inserted into the genome of a QRD2 expressing cell line, exposed the cell line to tamoxifen. And what we saw on the agarose gel was after tamoxifen exposure, this typical ladder effect, where each band corresponds to one of the sizes predicted by our model. And we saw that this recombination was really fast and efficient and ran even after to completion after a couple of hours. So encouraged by these results, we decided to make a in vivo lineage tracing mouse and called it LOX code mouse. And just to clarify something in the notation for later slides. So in this case, we have one element that is has been inverted to recombination, and we will denote this as a -7, while a barcode that has, say, some elements that are in the normal orientation, we just denote them by the numbers in the original position in the LOXP cassette. And if the element has been inverted, we add a minus in front. So this barcode would be 1, 2, -5. The LOX code technology is similar to an existing technology, which is called PolyLOX and has been published in 2017. However, there's some important differences to PolyLOX, and main differences are that PolyLOX has 9 elements, code elements, while LOX code has 13 elements. And also the LOXP sites are in the PolyLOX system 200 nucleotides apart, while in the LOX code cassette there are 8 to 14 nucleotides apart. So this has quite significant consequences on performance. So while the diversity of PolyLOX is 1.8 million barcodes, theoretical diversity, the authors reported they were using 2.5 microgram tamoxifen to get efficient barcode generation. And because the PolyLOX cassette is relatively long, you need PAC biosequencing to read out the full barcode. However, with LOX code, because we have more elements, we can generate higher diversity, about 15,000 times more barcodes in total. We can, because the LOXP sites are very close together, the cassette is more sensitive to tamoxifen, so we can use lower concentration of tamoxifen. And because the LOXP, LOX code is shorter in total length, we can use Illumina MySeq sequencing for the readout. So as a first challenge for this technology, we decided to test it on mouse embryogenesis. And for that, we collaborated with the lab at WeHi, the Taudi lab, and especially Christine Bivin. Embryogenesis is a highly dynamic process, starting from a single cell at the time of conception to about 200 epiblast cells at day 5.5. And the complete embryo, just 1 week later, with millions of cells, different tissues and structure. Sorry. So if you want to do lineage tracing in this system, there's obviously no point in barcoding the first cell because we know all the cells will be derived from it. Similarly, barcoding at the end of the development also is not very informative because the tissue, the cells have already differentiated into the various tissues. So we decided to barcode at this stage where we have about 200 epiblast cells, and then harvest the embryo one week later. The experimental setup of the data I'm going to show today is pretty straightforward. So we crossed the LOX code mouse with the ROSA 26 CREAT2 mice. After 5.5 days after plaque is detected, we inject tamoxifen into the mother, and then we harvest the embryos 1 week later at day 12.5. Then from these embryos, we have 3 different readouts in terms of barcodes. So we have PCR-based readout we have a single cell RNA-seq readout, and we have a spatial RNA-seq readout. Today I will talk about PCR and single cell RNA-seq. Before I move on, I would like to give you an overview of the different tissues we are sampling from the embryo. So we take brain tissues, neural tube tissues, several organs, face, and in addition, some mesenchymal tissues that are not shown on this slide. So let's start with the PCR-based readout. So we start with the embryos that we have collected. We have barcode a week before, and so embryos are dissected. They are lysed. This all happens in like a 96-well format, so we can process a large number of samples at the same time. We do a LogsCode PCR and library prep, and we sequence the barcodes on the MySeq platform. And this basically gives for each tissue, gives us the information about the clonal composition, so which barcodes are in which tissue and at what frequency. And for data I'm going to show today, we have processed 3 embryos and in total 288 samples. With this data, we can do what we call clonal correlation analysis, where we look at different barcode repertoires in the tissues and look at how they are related. Closely related tissues are expected to have more similar barcode distribution. And that's what we see in the data. So similar tissue types, like here for legs and limbs, are grouping together, brain tissues are grouping together, and so on and so forth. This is now based on hierarchical clustering, and as it's a heat map, we can just show one dimension, but obviously an embryo has 3 dimensions. So we tried a different visualization where we use a dimensionality reduction technique called UMAP, and which is very commonly used in, for instance, single cell RNA-seq data. But rather than using gene counts, here we're using barcode counts over tissues. So each dot is a different tissue, and we projected the barcode information onto those onto 2 dimensions. And so what we see is that again, the brain tissues are neural tissues are clearly grouping together. Also, left and right tissues are usually similar. Then we have the mesenchymal tissues grouping together. We have the forelimbs and hind limbs grouping together, and then the internal organs as well. So while this is a representation that doesn't require any input in terms of anatomy of the embryo, we also tried out a different visualization where we actually put in the knowledge we have about the position of each tissue in the embryo. So here are shown all the different tissues that we sample, and they are laid out in an anatomical map representing their position in the embryo. And we're connecting single nodes if they are highly correlated. If we split this up into different tissue types, like neural tissue, mesenchymal, and dermal-derived tissues, limbs, and gut, we see that they have very different patterns in terms of connectivity. So far, all the data I have shown so far is based on all the barcodes we find in our data. However, the LOX codes, some LOX codes are more easily made than others, so are more frequent. So there's a chance that they are made in more than one cell initially when we barcode the embryo at day 5.5. And so if we want to have confident information about individual clones, we need to remove those barcodes that are more frequent. And so while I don't have time to go to the details, I just want to show you the result in terms of filtering. So on the left, on this Venn diagram, we have shows the barcode we find in 3 different embryos, and this is basically showing that we get about 200 to 300 barcodes per embryo, and there's some overlap between the barcodes, which means that the same barcode was made in 2 different embryos. However, after filtering, where we remove more likely barcodes, we see that we still retain 35% of our original barcodes. However, now there's very little overlap between barcodes, suggesting that we can be confident about our data and single cell tracking. So then we can look at individual clones, and we can ask the question in how many different tissues we find this clone. And so what we see is that while a lot of clones are just found in a few tissues, some clones are found in most tissues. And when we look at biomass, those clones that are found in most tissues also represent the biggest contributor to the embryo. And if we plot the number of tissues a clone is found in versus its biomass, we see a clear correlation. So that just means that bigger clones are also found in more tissues. We see a broad range of the number of tissues a barcode is found in, and also a very broad range between the size of the different clones. So that means single epiblast cells produce a large variation in terms of their output. So we can also look at now at individual examples, and so here I'm coloring different tissues based on their frequency of barcode we find a specific barcode in. So in this case, it's an inverted 9, 8, inverted -1, 10, and 11 barcode. Remember, these are the original numbers in the cassette, but after recombination. And we see that this barcode is absent in the brain, while this barcode is present only in neural tissues. This is a multi-outcome clone, and this is a clone that has just been detected in a single organ. While we can look at individual barcodes, a more high throughput way is to again use dimensionality reduction technique. But this time, instead of having the barcode counts as input, now we use the frequency in different tissues as input. So each dot in this plot is an individual clone, epiblast clone, where we are confident that it's derived from a single cell. And so we see different patterns in terms of their tissue output. And we see about 5 different clusters. So for instance, this cluster, which corresponds to this cluster here, just is biased towards brain tissue and neural tube tissue. While this cluster seems to contribute to most tissues in the embryo. This cluster is absent in arm, forelimbs, and limbs, and so on. So we see different output of epiblast fate in this data. However, what we don't see is, of course, different tissues are composed of different cell types, so we can resolve those cell types with the simple PCR approach. So that's why we decided to move on with single cell RNA-seq. And so because the LOX code cassette is transcribed by the ROSA26 endogenous promoter, we can basically extract LOX code information with the single cell RNA-seq data. So again, similar to the previous protocol, we start with an embryo, we dissect it. Now with single cell, make single cell suspension, run it, multiplex the samples, run them on their Chromium X controller, generate cDNA, and generate gene expression libraries, but also LOX code libraries using a target amplification. And because we have a large number of samples, we use multi-seq multiplexing to process the same, from the same embryo, about 40 different tissues at the same time on the same day. So these are TSNE projections of barcode counts for 4 different pools that will run on 4 different lanes. And by this, we can identify the tissue origin of individual cells. We can put this all together and generate this UMAP and annotate the different clusters we find. We can also look at, because we have the tissue of origin information as well from the multiplexing, we can also look at how different cells from different tissues project onto this master UMAP. And we see certain regions like these are clearly enriched in neural cells, while these are more organ-specific cluster, and here we have the cluster for limbs and limbs. But now we have, in addition, the log-scode information. And so here I'm showing 4 different examples of log-scodes that we can detect single cells on this same UMAP. And you can immediately see that, again, here are shown the different elements that are contained in this log-scode. And as you can see, that the distribution is quite distinct. So there's some heterogeneity in transcriptional fate outcome of single clones, which recapitulates what we have seen from the bulk data, from the PCR-based approach. So now we have tissue information, we have transcriptional cluster identity, and we have log-scode information. And if we put it all together, we can generate this kind of contingency tables where we look at fate outcome of individual clones. Again, we can see that they are quite distinct in this case. And so interestingly, what we saw that individual clones tend to be in the same transcriptional cluster across different tissues that we sampled. So basically, there's a tendency of clones to make the same cell type, or they will be in a different tissue. However, at the same time, we also see that clones can make the same, can be in the same tissue, but make different cell types. So we kind of see both possibilities. Yeah. So we can now zoom into individual tissues, and in this case, an example is gonads, and we can look at which clones we find in this tissue. And so in this case, we have 12 different clones, and we can see here color indicates whether the cell was in the left or the right gonad. And we can see this clone, for instance, this barcode, was just detected in the right side, the right gonad, while this clone was just found in the right gonad. So we see some heterogeneity in outcome between left and right. Other clones have a more mixed outcome. So the question what we are interested in here, so we see clearly some heterogeneity, but we wanted to know whether single clones have some unique characteristic to them that makes them different from other clones. And so the answer to this question is yes, but let me explain. I need to explain a new concept, which we call the nearest neighbor clone, to understand these results. So this is an example of a clone in the gonads again. And so that's the original clone. And the nearest neighbor of a cell is basically defined as a cell that is as similar as possible to the original cell. So in this case, this would be the nearest neighbor cell. We can repeat this for each cell of this clone, and this would be basically define the nearest neighbor clone. And so now, if we compare each clone to all the other clones we have detected in the same tissue, we can ask the question, how different are they within clones? And the way we do this is by measuring the differentially expressed genes we find between clones. And so we did this comparison with the real clones we measured, but also we did the same comparison with the nearest neighbor clones, which are a set of cells that is that is as close as possible to the original clone in terms of transcriptome, but not clonally related, or unlikely to be clonally related. And we saw that we find clearly more DE genes in the case of the original clones, or the real clones, rather than the nearest neighbor pseudoclones. We can repeat a similar analysis for all the tissues. We see again that there's clearly more heterogeneity between clones, between real clones, and between nearest neighbor clones. So what does this mean? So apparently it seems that clones have some distinctive features that are not captured in that we can see on the normal UMAP representation, but that makes individual clones unique. So when this feature appears, or whether they have a biological significance, we are still exploring. So this brings me to my summary. So in terms of log-scode technology, I hope I have convinced you that log-score technology is a powerful new tool to do in vivo clonal lineage tracing in terms of embryogenesis and epiblast fate. So I've shown you some visualization of tissue relationships using clonal correlation analysis. We saw really huge variation in terms of output of clones, in terms of their tissue output between clones. And we saw intraclonal pattern of clones making the same cell in different tissues and in different tissues, and in the same tissue as well, different cell types. And I also discussed briefly the interclonal heterogeneity of some unique features that make clones different. So with that, I would like to thank everyone involved in this project. So especially my mentor, Charlene Naik, everyone in the Naik lab at WeHi, past members, Denise Marles for the cloning effort, and Stephen Sang for the analysis pipeline. It's also like to thank the Taudi lab for their involvement in the embryogenesis work, SCORE at WeHi for sequencing and a lot of support, and also funding bodies and support. Thank you very much. Happy to take questions.
Thanks, Tom. Tom, we might hold the questions till the end of the session, if that's okay. Perfect.
Sure, no problem.
Great. Next, I'd like to take this opportunity to introduce Professor Gabrielle Bells from the Diamantina. She's very familiar to this audience. So Gabrielle will be talking about some of her exciting single cell work in the gut. Over to you, Gabrielle.
Thanks, Arutha. I'll just share my screen. So I'm just going to give you a really short tour de force of what we're doing with looking more deeply at what's happening in the gut and homing down on individual cells. So we've got a real interest in understanding what individual epithelial cells in the gut and other tissues are actually doing. And there's, in different ways, been quite a lot of work done around this. More recently, there's been a huge amount of work done around enterocytes and how they might actually inform immune system responses. So we've been really interested in a couple of different types of cells, and they're tough cells and M cells. And so you can see in this example of what gut epithelium looks like, in fact, instead of just being a single type of cell, there's at least 6 or 7 different types of cells. And extending from what we've probably really previously thought about, they're not just a simple continuous structure of epithelium that's separating us from the microbiome and the internal parts of the body. They're actually operating in driving physical barriers, sensing of what the external environment is doing, together with transducing signals into the body, often through immune cells. And so this posed a really great challenge to us because some of these cells are incredibly rare. And while we know that they do really important things, actually having the tools and techniques that allow us to home down on those individual cells and start to understand exactly how they're communicating with other cells has been a real challenge. So this just shows you a Swiss roll of the small intestine of a mouse. And the yellow cells are really highlighting 2 different types of specialized epithelial cells. And they're so incredibly rare that they're actually really difficult to see even on immunohistochemistry. What we did to try and get some insights into what some of these cells were doing was create reporter mice. And so this is driven by a very specific promoter that allows it to be expressed principally in the cell types of interest. And you can see that there's this nice staining of these cells. If we dig a bit deeper on that and start to say, well, how many of these cells are there really and what do they look like? I mean, these are the sorts of cells that we're trying to understand their function of. So in this right-hand panel, you can see there's actually 2 different types of cells, the ones that are shown in pink and the ones that are shown in yellow. And so the challenge is that they share part of their transcriptome, in fact. And what we want to do is separate them out and then understand how they actually transduce those signals from the gut epithelium into different types of immune cells. And so I think the single cell technology and the 10x technology has really given us a new way to probe our cells and new ways to actually look at our cells. So using those fluorescent reporter mice, we were able to actually identify or isolate the epithelium from different tissues and then sort those cells so that we could investigate the transcriptome of those cells. And so this just shows you the UMAP of the clustering of those cells. So very nicely, we've got different populations of cells from really just a handful of cells that we've used as the input to this particular type of experiment. If we look at the transcriptome though, it's clear that there's considerable amount of diversity amongst these cells, not only in the different types of cells, but in the maturation states and potentially the differentiation of these different cells. And so this is all new information that really flies in the face of what we've historically known about some of these cell types. If we look in a more expansive way at these cells, you can see that there's really a huge diversity of cell types. And so these technologies that we're talking about today with 10x and the Visium, we're really trying to understand not only those individual transcripts, but we're trying to understand how those cells fit together, particularly when we're thinking about the immune system. Because we think that the immune cells at the barrier surfaces are really forming these sensory hubs. And while they do protect the body from infections, they're actually receiving micro pieces of information on a moment-by-moment basis so that they can integrate that information and tell immune cells what they should be really doing and exactly how much they should be getting activated or producing cytokines. What's interesting about homeostasis is it's that feature of the immune system that stops you being sick on a day-to-day basis, despite the fact that we're exposed to trillions of bacteria and insults every day. So we've actually taken our single cell data and we've tried to interrogate that data with respect to what physically these cells would look like and how they're anatomically positioned with other cells. So this just shows you a three-dimensional rendition of some imaging that we've done. And so what we see in the epithelium is that the blue cells are your average epithelial cell. And then we have M cells and tuft cells. And they form interactions that were not previously thought to occur. Sometimes they occur as single cells, sometimes they're occurring as clusters. And we think they've got feed forward and feedback mechanisms that are really important for homeostasis. This by itself is a really nice image, but it's quite static. And so the challenge for us has been to integrate the single cell data with this sort of very nice visual data that gives you a feel for where cells are actually localized. So single cell data has, there's many ways of interrogating that sort of data. It's often quite complicated, but there's some really nice platforms for having a look at that data now. And so for us, we wanted to know how closely associated each of these cells were to other types of immune cells. So what is the epithelial cell identity related to the immune cell identity, and what receptors and ligands might be driving that? In the past, most of these epithelial cells have been thought to actually interact with dendritic cells. So using this particular type of modeling together with Matt Ritchie's lab and Yu and Wang Ko, who's PhD students in each of our labs, we've been able to tease apart the probability that different receptors in our mix of cell transcriptomics would be reflecting the localization of those cells. And it's quite interesting in that most of the localization is not necessarily by dendritic cells. It's by these myriad of other cells, such as gamma delta cells, innate lymphoid cells, and tissue resident T cells that are found in the lamina propria that are forming this sort of battalion at that frontier surface. So that's just been a very brief synopsis of what we're doing, but I think these technologies really allow us to integrate a vast amount of information that we're now able to glean from very, very rare populations of cells, which is the space that we operate in most of the time, and integrate them three-dimensionally to try and understand what the integrated physiology of those tissues are. So with that, I would thank everyone who's participated in this research, which is mainly driven by both Matt and my PhD students, so Wang Ko and Yu. And we've had a huge amount of support from funding bodies and particularly from the people who are developing all these technologies. We really look forward to applying some of these techniques to some even more challenging tissues, such as lung tissues. So thank you.
Thank you so much, Gabrielle. So I'll be closing out the launch today, and I just wanted to, first thank all of our speakers, invited and local speakers today. I think we had some really great talks, and definitely some highlights of what you can leverage 10x technology to, the type of data that you can generate from 10x technologies. But also, first and foremost, I wanted to thank Arutha and Gabrielle for their support in launching this program at UQDI. When I first talked to them about it, that we recognized that that something that was lacking was some strong genomics capacity at UQDI in the context of single cell technologies. And one of the major focuses of this program is about enablement. And then on top of that, we built this education program to go with it. And so we really think, through this enablement education, we can really help advance the research at UQDI as well. So, with this, now I just wanted to thank everyone for attending, and we'll open up the floor to questions to all of the speakers now. So, and I'll also be happy to answer any questions related to the program. Yeah, thank you.
And we've got
Sophie, I have your application. We'll just run it through the program now. So no need to reapply.
There's a question from Joseph to Jeff, who is asking, is the Visium assay compatible with all tissue fixatives?
Yeah, good question. So at the moment, there's two versions of the kit. There is the fresh frozen, so there's no fixative involved in there, and the tissue needs to be embedded in OCT. And then the other version of the kit, which is FFBE, so it's formalin fixed. That's the only fixative we're compatible with at the moment. However, please reach out and discuss your use of other fixatives, if they could be compatible, how we might map some of your existing tissues if you've got them stored, or at least the upcoming storage conditions onto the actual slides themselves. It would be good to better understand what you're doing and if there's any path forward there.
Yep. Karen, can you unmute Habib has a question.
Then I had a question.
Sorry. Go ahead, Ruth. I think Karen, did you hear me about Habib? Oh, there we go. Karen's got it. Habib, you can just unmute yourself and ask your question.
Hey guys, no, I don't have any questions. Thanks.
Oh, okay. Thank you. All right, go ahead, Arutha.
Yeah, Dan. For the single cell workflow that's being supported at DI, is there scope, I guess, for the Visium workflow to be established here as well, and sort of developing that?
Yeah, certainly. So the fellowship program is not limited to single cell. We are also including spatial and enabling spatial training and enablement of that, establishing that workflow at UQDI.
Yep. Thank you.
I think Fiona had a question. Fiona, you might elaborate. Best, are you asking what the best fixation? I'm a little bit unclear of the question. Can you ask the question in a different way, Fiona?
I'll try and answer it, assuming I know what Fiona was asking, but is it best to use fresh frozen tissue, or is it best to use the fixed platform? I hope that's kind of what you're asking. It might get clarified in a second in the comments, but the answer to that question is, which assay is better than the other. If you've got recently fixed tissue, then we find through our in-house data, the performance of that assay is equivalent to the fresh. If you've got tissue that's, say, being fixed for longer periods of time, we have some upfront QC to perform on that to determine basically what expectation to set out of that tissue. We do find sort of older blocks, maybe 10 years and older, do degrade the performance of the fixed assay, but for reasonably fixed blocks, the performance of the fresh versus the fixed assay is equivalent.
There were some follow-up questions on that about that Fiona's got tissues with perineal invasion from head and neck that's frozen. And if we wanted to map nerve versus tissue versus tumor.
Yeah, we've already got them frozen. Depends how they're frozen, if they've been snap frozen or we've got particular freezing recommendations, but certainly if you've already got them frozen, they'll be compatible with the fresh frozen version of the kit. Keep in mind the resolution being 55 micron. If you're interested in boundaries that have single cell layers, then the Visium HD might be applicable to what you're doing as opposed to the current iteration of the product. But most certainly if you've got already frozen material that's compatible with the fresh frozen kit.
Jeff, I might go back to the initial question from Anne, and just so you can answer that here, for studies on the microbiome, is there a way to use bacterial DNA probes with the Visium spatial technology? And I just know there's definitely a few interested at UQDI that are interested in this capacity. So maybe you could answer that for everyone.
Yeah, definitely. We're going to actually get a lot of questions about this, either bacterial or virus particles, can we detect them? So for the fresh frozen version of the assay, we detect any polyadenylated transcripts. So if your bacteria or other non-cell species have polyadenylated transcripts in them, then they should be picked up by the assay. Then it's just a matter of aligning those transcripts to the transcriptome that you've got and then searching for what you're looking for. For the fixed version of the assay, we actually use a probe-based detection system. So we don't detect the transcripts directly. We actually use probes that bind to the transcripts, and then we barcode those probes. Because we're using probes, we have a defined probe set, and that defined probe set covers about 18,000, or essentially all the protein coding genes across the transcriptome. So that probe set won't pick up anything other than what we've already designed it to do. So the short answer is no, basically, you can't detect it, but there are always ways in protocol hacks, if you will. I think a particularly successful group in New South Wales has actually added polyadenylated regions to DNA oligonucleotides. They were doing this for the purpose of detecting other species, but there are always pathways forward of adding particular things into the species you want to present so they can become visible to the assay. Do know that those kind of approaches typically aren't supported by 10x, meaning it's difficult. We don't have software for it, and some of the workflow changes might change our product performance. But nevertheless, we're always happy to talk through what you're trying to detect and see if we can feasibly map it across to a product.
Yeah. All right, thanks, Jeff. We might wrap it up there. I just once again wanted to thank all of the speakers. I think we, like I said, it was a great technical talk from Jeff, and great perspective on use of the technology from Gabrielle and Tom. I just wanted to remind everyone again that right after this event, there's the 10x Genomics Global Experience event. A few things that we didn't really highlight today that are coming in the pipeline that are very exciting. We have our fixed RNA profiling kit, which lets you do single cell gene expression on fixed single cells suspensions. Co-detection of protein with Visium, so on fixed tissues, along with our upcoming in situ platform that Jeff highlighted. So some really exciting stuff coming. So highly encourage you to attend the experience event. So yeah, I just want to say that we're all very excited to launch this program at UQDI, and we're really excited to see the data that comes from it. So thank you all.
Thanks, Dan.
Yeah, thanks, Aretha. Thanks, Gabrielle.
Thanks, everyone. And yeah, thanks really for driving this here. I mean, I know people are excited. Maybe that's just me.